Chapter 3A.1.2 How Neurons Communicate. How do nerve cells communicate with other nerve cells? All information processing in the brain involves neurons talking to each other at synapses, says neuroscientist Solomon H. Snyder in 1984. Neurons interweave so intricately that even with a microscope you would have trouble seeing where one neuron ends and another begins. Scientists once believed that the axon of one cell fused with the dendrites of another in an uninterrupted fabric. Then British physiologist Sir Charles Sherrington, 1857 to 1952, noticed that neural impulses were taking an unexpectedly long time to travel a neural pathway. Inferring that there must be a brief interruption in the transmission, Sherrington called the meeting point between neurons a synapse. We now know that the axon terminal of one neuron is in fact separated from the receiving neuron by a synaptic gap, or synaptic cleft, less than a millionth of an inch wide. Spanish anatomist Santiago Ramón y Cajal, 1852 to 1934, marveled at these near unions of neurons, calling them protoplasmic kisses. Like elegant ladies air kissing so as not to muss their makeup, dendrites and axons don't quite touch, notes poet Diane Ackerman in 2004. How do the neurons execute this protoplasmic kiss, sending information across the tiny synaptic gap? The answer is one of the important scientific discoveries of our age. When an action potential reaches the knob-like terminals at an axon's end, it triggers the release of chemical messengers called neurotransmitters, figure 3.4. Within 1/10,000th of a second, the neurotransmitter molecules cross the synaptic gap and bind to receptor sites on the receiving neuron, as precisely as a key fits a lock. For an instant, the neurotransmitter unlocks tiny channels at the receiving site, and electrically charged atoms flow in, exciting or inhibiting the receiving neuron's readiness to fire. Then, in a process called reuptake, the sending neuron reabsorbs the excess neurotransmitters. Chapter 3A.1.3 How Neurotransmitters Influence Us. How do neurotransmitters influence behavior, and how do drugs and other chemicals affect neurotransmission? When it comes to the brain, if you want to see the action, follow the neurotransmitters, says neuroscientist Floyd Bloom in 1993. In their quest to understand neural communication, researchers have discovered dozens of different neurotransmitters and almost as many new questions. Are certain neurotransmitters found only in specific places? How do they affect our moods, memories, and mental abilities? Can we boost or diminish these effects through drugs or diet? In later units, we will examine neurotransmitter influences on depression and euphoria, hunger and thinking, addictions and therapy. For now, let's glimpse how neurotransmitters influence our motions and our emotions. A particular pathway in the brain may use only one or two neurotransmitters, and particular neurotransmitters may have particular effects on behavior and emotions. See Figure 3.5 and Table 3.1 for examples. Acetylcholine, ACH, is one of the best understood neurotransmitters. In addition to its role in learning and memory, ACH is the messenger at every junction between a motor neuron and skeletal muscle. When ACH is released to our muscle cell receptors, the muscle contracts. If ACH transmission is blocked, as happens during some kinds of anesthesia, the muscles cannot contract and we are paralyzed. Physician Lewis Thomas on the endorphins. There it is, a biologically universal act of mercy. I cannot explain it except to say that I would put it in had I been around at the very beginning, sitting as a member of a planning committee. The Youngest Science, 1983. Candace Pert and Solomon Snyder, in 1973, made an exciting discovery about neurotransmitters when they attached a radioactive tracer to morphine, showing where it was taken up in an animal's brain. The morphine, an opiate drug that elevates mood and eases pain, bound to receptors in areas linked with mood and pain sensations. But why would the brain have these opiate receptors? Why would it have a chemical lock unless it also had a natural key to open it? Researchers soon confirmed that the brain does indeed produce its own naturally occurring opiates. Our body releases several types of neurotransmitter molecules similar to morphine in response to pain and vigorous exercise. These endorphins, short for endogenous, produced within, morphine, as we now call them, help explain good feelings such as the runner's high, the pain-killing effects of acupuncture, and the indifference to pain in some severely injured people. But once again, new knowledge led to new questions. How drugs and other chemicals alter neurotransmission. If indeed the endorphins lessen pain and boost mood, why not flood the brain with artificial opiates, thereby intensifying the brain's own feel-good chemistry? One problem is that when flooded with opiate drugs such as heroin and morphine, the brain may stop producing its own natural opiates. When the drug is withdrawn, the brain may then be deprived of any form of opiate, causing intense discomfort. For suppressing the body's own neurotransmitter production, nature charges a price. Drugs and other chemicals affect brain chemistry at synapses, often by either amplifying or blocking a neurotransmitter's activity. An agonist molecule may be similar enough to a neurotransmitter to bind to its receptor and mimic its effects. Figure 3.6b. Some opiate drugs are agonists and produce a temporary high by amplifying normal sensations of arousal or pleasure. Not so pleasant are the effects of black widow spider venom, which floods synapses with ACH. The result? Violent muscle contractions, convulsions, and possible death. Antagonists also bind to receptors, but their effect is instead to block a neurotransmitter's functioning. Botulinum, a poison that can form in improperly canned food, causes paralysis by blocking ACH release. Small injections of botulinum, Botox, smooth wrinkles by paralyzing the underlying facial muscles. Other antagonists are enough like the natural neurotransmitter to occupy its receptor site and block its effect, as in figure 3.6c, but are not similar enough to stimulate the receptor, rather like foreign coins that fit into but won't operate a soda or candy machine. Curare, a poison certain South American Indians have applied to hunting dart tips, occupies and blocks ACH receptor sites, leaving the neurotransmitter unable to affect the muscles. Struck by one of these darts, an animal becomes paralyzed. Before you move on, ask yourself, can you recall a time when the endorphin response may have protected you from feeling extreme pain? Test yourself. 1. How do neurons communicate with one another?